Our next presentation will be the challenges of challenge studies with Dr. Jason Bolton, Food Safety Specialist from the University of Maine, and Mary Yavelak, Food Safety Associate at North Carolina State University. This is a pre-recorded video with a live Q&A at the end. Mary will be answering your questions, so please post them in chat. Amy, could you please play the pre-recorded video?
Hi, everyone. My name is Mary Yavelak. I am an Extension Associate at North Carolina State University and also the Retail Program Manager for the Safe Plates brand at NC State. Jason, do you want to introduce yourself?
Sure. Jason Bolton, Food Safety Specialist, Extension Professor, and I run the Innovation Center out of the Maine Business School at the University of Maine.
And today we're going to talk to you about the challenge of challenge studies. So first, let's talk about what challenge studies are. Challenge studies are a source of validation. Typically in retail, we're looking at extended hold times and treating foods as non-TCS. And so this is just the validation study that goes along with that. It's important to note that challenge studies are specific to the food and the process. So it's going to go through the entire process that's being done at that retail establishment for that food and determine a safety parameter around that. And so if it's done for other foods or other processes, it needs another study. And we'll talk about that part a little bit later. Intrinsic properties of food is really important here. So that's things like pH and water activity. And that's really what the food code uses to determine if foods are TCS or non-TCS. The tables on the slide are taken straight from the food code. Table A deals with foods that are heat treated and packaged. Table B deals with foods that are not heat treated or heat treated but not packaged. And so you can see that there are pH and water activity values that, decide if foods are TCS or non-TCS, but there are other factors that need a product assessment. And that's where a challenge study can come in and kind of give us some more information about some of these foods that are kind of ambiguous in the food code as to how they should be handled and whether they can support pathogen growth. The other thing that's really important with challenge studies is that they're not always needed. So challenge studies can be an expensive expense for a establishment. And like I said, they give an external look at what's happening at a point in time, replicated a very well-designed study, but if it is a product that is, they're going for a variance, they're needing a HACCP plan for something like sushi rice or a more simple process, they may want to use a HACCP plan, a variance, and do monitoring as part of that HACCP plan, and use that to determine, if they can hold this food as a non-TCS food. The reasons that that can be used instead of, going through this challenge study process. Number one, it can be more cost effective. It also gives some real-time data in terms of, say sushi rice is being made every day at an establishment. It gives a look at what that pH is on that day and, gives some corrective action abilities there. But it's also important to note that that's the monitoring piece can be done for some foods, like I said, sushi rice, yogurt, fermentation, but is not applicable to everything. And that's kind of where challenge studies can come in. An establishment who, does a lot of different types of food may not have the money, may not have the time, the expertise to do water activity monitoring, those kinds of things. And so that's where a challenge study can come in and kind of bridge that gap, give that good analysis of what's happening in this process and, decide the properties of whether it can be held as they want it to or not. But we did want to make sure we mentioned that it is not necessary to do a challenge study for every food and that sometimes in-house monitoring can be done in some of the establishments that you'll see. The other thing that we wanted to mention is that there are validations and challenge studies that already are in the literature. And they're published data that can be used either in lieu of performing a new study or even kind of to supplement study design, trying to figure out pathogens of concern and those kinds of things. When using a validation and challenge study that's already occurring in the literature, it's really important that the properties of the food are the same or at least similar enough. Those intrinsic properties might be the pH of the food, the water activity, any antimicrobial properties that the food has that would interact with the pathogens of concern there. Also, when we're talking about lethality, size and thickness really matter. So when we're talking about like this validation resource to the right on this slide is one for beef jerky, and it does mention a specific size and thickness of the strips. So if an operator did want to use a validation that was already published, it's really important that they don't stray from the process that was studied during this challenge study. And if they do want to stray from it, it's going to need a new challenge study to occur. So, it can be kind of daunting for an operator to try to figure out where there might be validations and challenge studies already in the literature, if they're applicable to their process. And that's where it's really important to know, and we'll talk about it multiple times during this presentation, that there are extension regulatory contacts near you at a local level that can help find this published data, can help determine if it's applicable to the situation. So if an operator is wanting to, find something that's already out there, it's important to contact local regulators or state regulators, retail specialists, or your university or extension, food science, food safety contacts, and kind of start that conversation as to whether it is an appropriate use in that establishment before they go ahead and do that. So there's really three parts to these challenge studies that we'll talk about, and we're really giving you more of an overview today on challenge studies. The first part is designing challenge studies, and a lot of the information from this presentation was actually taken from the NACMIF document. We'll reference it a couple times during this presentation, but that is, if you're looking for a little more in detail than what we can provide in the time we have today, that's a great reference to go and look at. So recommended expertise for somebody who's going to design these challenge studies is an experienced food microbiologist. The considerations that they may need to have in this is the intention of the study, and that's really to make sure that the pass-fail parameters that are coming out of this study are matching what the operation wants to do, because you wouldn't want to go to the end of this procedure that's expensive, it's time-consuming, and then find out that you have some limits that don't really meet what the operation wants to do. It's also really important to identify the pathogens of concern in the food product, and we'll talk about that a little more on the next slide. Microbial models can be used in challenge studies, but they're typically used in the design phase, and a lot with determining the pathogens of concern. They can be used to determine growth there and figure out kind of which ones to focus on in the challenge study. Also, like I said at the beginning, this challenge study is going to follow the exact process that the food goes through in the establishment. And so if there's any other processing techniques, the specific packaging, all of that needs to be considered in designing this study so that it is as applicable to the establishment as possible. We wouldn't want something to happen in a lab that then cannot be taken to the food establishment to actually be implemented. The other part is that these challenge studies, are eventually going to be looked at by a regulatory body. And so involving them in the design phase can only be, helpful, can only, help the operator, the regulator kind of figure out if this is going to be something that is approved at the end and approved for use. A worst case scenario would be having to kind of go and do all of this, and at the end it not be accepted. And so the labs that are chosen for these challenge studies may have the expertise, this experienced food microbiologist to do this, but they also may not. And so I've already mentioned that there are university and regulatory and extension contacts that are in your state that can be used, if a lab that is going to be used by an operator for a challenge study doesn't have, that experience. It's more important to kind of reach out and figure out and have somebody with that expertise rather than to kind of just go with a lab and go with kind of what they have in hand. Jason, I think you have a story for us.
Yeah, I mean, so I've experienced a few large commercial, but at a retail level type challenge studies, and because one of the organizations that I work with didn't hire a lab that really understood this general process and understood starting concentrations or loads of bacteria, certain pathogens of concern, and also appropriate AOAC methodology or the latest methodology, companies spend a lot of money on a challenge study only for it to be turned down when experts take a look at it. So that's where, what Mary said, really important to have the expertise and multiple people looking at this, not just relying necessarily on the good word of the lab. So really making sure that you do your due diligence before you invest in what can be, tens of thousands of dollars for an appropriate challenge study.
And in determining the potential pathogens of concern, so this table is actually taken from that NACMF document that I've referenced, and it can be useful in determining potential pathogens of concern based on pH and water activity. Obviously, there are typical pathogens of concern in, every food product. And so it's important to kind of cross-check this with lists, to determine which pathogens or surrogates or whatever is appropriate to use during the study is used. But this list gives kind of a starting point of the intrinsic properties of the food and what may be able to grow based on those properties. And then the list can kind of be kind of tailored down from there. Like, for example, Vibrio doesn't need to be tested in a large array of foods, so it's important to also look and not test for everything, but to figure out kind of from this starting point, which potential pathogens of concern should be enumerated and figured out in these studies, and when a surrogate is more appropriate for that.
Okay, so I'll talk a little bit about conducting challenge studies, and as Mary has mentioned, it really comes down to that expertise of not only the folks that are going to work for the company or the organization that you've hired, and that organization could be a private lab or it could be a university. So sometimes universities will perform challenge studies as a for fee or as part of a research project. We've done some stuff here at the University of Maine as a part of research projects where we've actually performed challenge studies for a particular type of product and or process. So we want to make sure that the folks that you're working with have an expertise in microbiology, that they have conducted these type of things before, and in some cases, maybe even a team. So when you're selecting the lab, that's something you want to look for. Also, it's helpful, in addition to that trained microbiologist, we talk a lot about microbiology here, but in some cases, it might be an aspect of a chemical analysis or any other type of property where you might be concerned about a particular hazard or type of hazard. So you want to make sure that they have that expertise with the type of hazard or hazards you're concerned with. And that also, in some cases, ideal situations, some of the larger labs will even have regulatory experience. It could be retired regulatory folks or folks that have left regulatory to enter into the private world. So that expertise and that experience can really be helpful. So do your due diligence before doing that and going out and buying or hiring a lab. In addition, if you're a regulatory body now looking at a challenge study, it is worth looking into what types of certifications and what are the credentials of those folks working the lab or running the study. So that's something that you can kind of look for. what type of validated methodologies. So typically they're going to be AOAC or referenced by some sort of peer-reviewed journal or research, so that there is a method that is actually something that's an industry standard opposed to something they've maybe made up within the lab. Testing the pathogens of concern and, when appropriate, those surrogates. Not always are the surrogates appropriate, but in some cases, depending upon the types of microorganisms that are present, as long as they're using a surrogate that's from a source that actually makes sense. So sometimes I've seen some studies where they've used surrogates and the surrogate was not appropriate for that particular product or process, or it hadn't been even validated for use in that area with that type of food. So something you want to really look at, but this is where it comes back to having an expert in food microbiology that can actually look at these things. And then the replication of experiments. Typically this is run in duplicate or triplicate. And so you want to make sure that that's actually been done. And it's been done really drilled down in the details of how they ran that. So not only the methods for looking at enumeration of the microorganisms, but also were they replicating what this organization wanted to do from a process point of view? Were the temperatures the exact same? Were the quantities, the materials, the processing equipment, whatever it may be? You really need to make sure that that aligns with what the organization is trying to accomplish. Because I've looked at challenge studies where they're doing one thing and then it was just different enough when they were actually executing it in real life that it really made it kind of invalid. So you want to look at all those details, and it is helpful to get that team together, someone who has the microbiology experience. Next slide, please, Mary. Thank you. So the analyzing of the data, this is where it comes back. I mentioned some of these things, but variations in the samples, you want to make sure that in some cases it makes sense where they're going to have to run statistical analysis on this, so that they run statistical analysis on this information or this data that actually makes sense. Were they running the types of analyses that make sense for this? I've seen that where sometimes you'll see that data and you'll see that they mentioned something and it actually was an inappropriate analysis, and so the data may not be valid in that way. Significance of the results are obviously important, but it's all based on variation that occurs within those samples and also the statistical analysis that they're running. Assessing growth versus toxin formation is going to be really important. So some organisms you're going to look at, okay, they might grow, and growth can tend to mean toxin formation. And so you want to make sure that you really understand the organisms that you're looking at. So some you're just looking at growth. Some it's important to look at, like, Clostridium perfringens or any of the Clostridium species. Really going to be expensive studies, because typically if you're talking about any type of toxin formation, you're going to need a level 3 biohazard lab to run those things. However, most microbiologists have ways of doing that without forming toxins, so they can do that in level 2 microbiological labs. We're going to talk a little bit more about the aspect of shelf life versus product safety, but that's going to be something that's really important. Sometimes folks will submit data that is more of a shelf life opposed to an actual challenge study, so we'll talk about that. But one of the things to really note is when you're looking at growth and things like that, typically less than a log increase of some sort of growth or anything like that is not necessarily significant, but that brings you back to the point of what did they inoculate with? How much of the microorganism did they inoculate with? And I've seen a lot of studies where they didn't inoculate with enough, or enough that you could actually enumerate after several days. So that's something that's really important, and it has to make sense for the type of food and the intrinsic properties of the food. So shelf life studies, I get this a lot where folks will say, well, I don't know if this food is safe, so I'm just going to run a shelf life study. Shelf life study is really around the objective measure of the quality factors, not necessarily the safety factors of the food. So it doesn't equate to food safety. It's as simple, how long is this food going to last before the quality of the food is no longer, and there are some standards that are no longer acceptable. So some of the standards could be generic, non-pathogenic spoilage organism growth, or color changes, or texture changes, and things like that. It can't be used in lieu or in substitute of a challenge study. So that's something that's really important. We get that a lot. I'm just going to run a challenge or a shelf life study. That doesn't equate to safety. So that's something that they should do, because they should know how long the food should last, especially if you're talking about certain retail establishments, but that's not something that's a substitute for validation or challenge. And then microbiological analysis will sometimes get questions around, well, I sent my food out for testing and it shows that there are no pathogens present. the presence or absence of pathogens from a microbiological, even if they ran multiple samples, doesn't necessarily equate to the safety of the food. It's not a substitute for a challenge study or validation. It's a nice spot check to know, even if they took a composite sample of a bunch of different samples into one test, that doesn't necessarily mean that the product is safe. It just means that that particular batch that they tested may not contain pathogens of concern, but it doesn't necessarily equate to the safety. So we never use these kind of spot checks or even multiple spot checks of microbiological analysis to say that this food is safe by any means. It's really a lot of commercial processors use microbiological analysis to just look at, or microbiological tests to just look at an added verification, but not a validation or the challenge study. And so there are a lot of different resources out there. We're going to point and focus on the university extension contacts in your area. And if there isn't someone in your area or in your state that has an expertise, because we know that there's not necessarily someone like this in every single state, then I would definitely reach out to neighboring states, or at least start with your local state contact, and they might have a contact they can reference. But that's really important. Other regulatory bodies, I know you all have a pretty good network of regulatory contacts, but looking to see what other people are doing in other states is really important. Reputable consultants. The consulting world is full of a wide range of people and experiences. And so you want to make sure that if you are an organization looking at a validation or a challenge study and you need help with those things and you're going to hire someone out, that you look at references from those reliable, from consultants to determine if they're reliable or not, or they have the expertise that they say they do. The document that Mary recommended earlier, a lot of information in that, and that's a great reference to start with. And if you don't have access to that or you need a link for that, we can provide that. You're welcome. We're going to give you our contact information. You're welcome to contact us. And so those are the things that are really, really helpful. But going back to that university or extension context, they're going to be fantastic resources for you. And then our contact information, and feel free to contact us. And if we can't answer the questions, I'm sure we can find someone who can. Well, thank you, everyone.
Well, thank you, Amy, and welcome, Mary. I know you're online.
Yes, I'm here.
Hi, Mary. Would you like to start some of the chat questions?
Yes.
I know there were some questions about NACMIS.
Yes, and before I do, I'm going to go ahead. There were a couple people who asked for the link, so I'm going to go ahead and drop a couple of links, and hopefully it doesn't get buried. There's a lot of people saying thank you, and so hopefully this doesn't get too buried, so you can scroll up if it does. So some of the things that I'm dropping in, and I'm not pressing enter yet until I finish, I'm dropping the link to that NACMIF document, and so that's that National Advisory Committee on Microbial Criteria for Foods. That's the Inoculated Pack Challenge Study document. It's like a lot of pages, hundreds of pages of protocols. And so it really gets into the depths of this more than we could talk about today. In addition to that, I'm dropping a link for the AFTO Process Authority list. And so that's a great place to start. By no means, if somebody is on this list, does it completely qualify them for, saying that they're an experienced person to hold these studies, but it's a good person to reach out to. It's a good way to find somebody local to you. And also that doesn't mean if somebody's not on this list, it doesn't mean that they're not a good person to reach out to. But it is a good place to start if you don't know where to start. And then also me and Jason's emails are on there. In addition to that, I did mention, I've mentioned the NACMIF document a couple of times, and there is a CFP guidance document that was accepted this year at CFP. So kind of the backstory on that is during the 2018 CFP meeting, somebody from industry submitted an issue that asked for standardization of product assessments and challenge studies nationwide. So the product assessment committee created a guidance document for that. And that's really going to help, you all at the, the retail the local level be able to use this. Operators, I saw somebody asked a question earlier about, if I'm an operator or if I'm a regulator, and, how do I know if these are validated? And so that product assessment checklist that was accepted this year on CFP, that will eventually be on the CFP website, will be a great tool for you all to use, in your jurisdictions to, kind of use this NACMIF document and utilize it more kind of at the local level.
Mary, we did have a question. It says, is a challenge study required for spice manufacturers if ASTA has already made recommendations on what treatment parameters will achieve a 5-log reduction for salmonella? Don't know if you would know that.
And so there are 2 different types of challenge studies that usually happen. There are like holding longer studies, so like non-TCS, and then there's also lethality. And so, if we're talking about 5-log reduction, there would need to be some sort of, validation that the lethality does happen there. But so there are those kind of 2 different things. So there is, different things like if you want to leave cheese pizza out for 8 hours, that's something where you're looking at the amount that could grow over that time. But then there's also looking at the lethality studies, and both of those are covered in that NACMIF document.
Thanks, Mary. There was another comment / question about seeing vendors looking at staff levels instead of pH and water activity.
Yeah, that is a great point. So, kind of go back to the first couple of slides that I had. So, pH and water activity tell us a lot about foods. And so using that table in A and B in the food code can really tell us a lot about foods without having to do extra, studies. If somebody wants to look at the, a staff level, because they have some, there's some information that says that the pH and water activity, may support the growth, then, that can be used. But it's not always needed if the pH and water activity are, showing that it doesn't support the growth of things. And so it's really, it's important to kind of understand that and kind of know those intrinsic properties. And that's really why it's important to, involve that experienced microbiologist, and they can kind of make sure that, a small operator isn't more or less strong-armed into, getting a study that they don't need, but that also the studies that are needed are being designed well and executed well. And I also saw just that, large story from Stephanie about the chain and, not being a regulator. I won't speak to, accepting or rejecting something based on, but, it does really drive home the point that we had on if things change in these processes, that the challenge study may need to change. If there are changes in brands or there are changes in the process, then, there either needs to be something that shows that it, is similar enough that there doesn't need to be another one, or, it needs to, have an additional study. And so thanks, Stephanie, for your story. That kind of drives our point home as to, why these studies need to be really designed well in order to make sure that we're, analyzing what we need to analyze, in order to make the recommendations we do.
Thank you, Mary. That was a very great presentation, and the video was very helpful. We really appreciate your time today. We know how busy both you and Jason are. We will now take a 5-minute break.